Hello again. Hello and welcome again. Oliver here, Microbehunter here, and welcome to another Microscopy Saturday Microscopy live stream. Yeah, I'm happy that you were able to join again. Like every week at the beginning of the live stream, I have to do a little bit of housekeeping as well. I have to see if I can see myself in the video. Yes, this seems to work. So the stream has started. And yeah, essentially, yeah, here we go. I think I'm online now. I see that many of you have already joined. Just want to say, like every week, a hello around the world to Vietnam, to Armenia, to the Netherlands, to the Belgian coast, to England, to the UK again, UK, Essex, UK, Florida, United States, Netherlands. Now I lost it where I was. Oh, Hamburg in Germany. Okay, Poland, Denmark. Sound is good. Thank you very much. That's important because that's already happened before that I started to talk and there was no video and no sound. Okay, from Finland. Okay, from the Philippines. Okay, so yeah, I thank you again for joining in. Today I want to talk about two things I was kind of thinking of. You see, every live stream is a little bit flexible. I only have a very rough program, so a little bit the content depends a little bit also on your input. And if you have any questions, okay, and if you have any, and I see that some questions already appeared, right? I'm going to do today again what I usually do is all off-topic questions I would like to answer after approximately 1 hour. Please do write all questions by writing at Microbehunter, so it's actually easier for me to find the questions or comments that are for me. Because yeah, sometimes people like to use the chat box also to communicate with each other, which is fine. That's great, but it makes it a little more difficult to find the questions that are for me. So please indicate at Microbehunter, and then I'm going to skip over those questions and I would like to answer them later. Hello from Connecticut, the United States. Okay, so two things that I want to talk about today is I want to talk a little bit about slide boxes. I've got a new system. I found a new one that I would like want to show you. You've probably already seen it on the thumbnail. And then the second thing is I want to put some honey under the microscope because just recently I bought some honey. You know, some nice honey. I'm just going to use a centrifuge to extract the pollen from the honey. Just want to show you how this actually looks like. Here, that's basically now I've set up another camera. So I'll be using again my centrifuge here. It's going to be a little bit noisy, but we can get some pollen out of the honey. And that's basically the second thing. But then again, it depends a little bit on what your interests are and if there are any questions from your side. Okay, so let's for, I don't know, 15 minutes, 20 minutes, I don't know, let's talk a little bit about slide boxes and the reason why I bought a new system over here. Right, and some good news and some bad news and some weird news, whatever. Right. So for many years I've been using this slide box over here, which is a high quality slide box. Some of you might already know that I started to collect some antique microscope slides, usually around 100 years old. Really nice specimens, and I put them in here into this slide box, and I've kept them in here for a couple of months, but then I took them out now and I changed the system, and I will tell you why. This slide box over here, it's labeled in German. Microscopic specimens is an order number here as well. I don't even remember where I got it from. This is a long time ago, 20 years ago I bought those in some kind of a shop. Really nice. It's made of cardboard here. Inside it's wood, some plastic. And these are really nice slide boxes. It's not slide boxes. You can close it here, right? However, there was a problem with that. And the problem is that some of the antique microscope slides were so thick. The slide was so thick that it wouldn't fit in here, right? And so that was really a somewhat unexpected. So some of them were maybe even twice as thick as some other slides, so I could not fit them in here. So that was actually one problem. But that was not the reason why I changed them. The reason is you should keep slides, especially antique slides, not vertically but horizontally. Because this way you're able to store them much longer, because if you keep a slide vertically, I'm just going to use a modern slide right now, That's basically, yeah. Then it can be, I read that over the years that it actually starts to flow down, especially if the mounting medium is not completely solid, right? So there are some tensions and some strains on here. So basically, yeah, that's the reason why, especially valuable slides, if you want to keep them for a long time, apparently should be stored horizontally. All right. So this is the reason why for the antique slides, and I'm going to show them to you in just a minute or so, I took them out and I changed the system. And what I've done is I bought myself online, and there was actually only one place that I could find those, let me put this away, those files here. This one is still originally packed, and I bought 5 of them. And this one is still plastic wrapped, so I'm going to unpack it together with you so that we can have a look at this. And it's made completely of cardboard. It's not quite as cheap, really. It's around paid around 10 euros for one of them here. So, yeah, I said it's why not? If this is what it takes to keep the slides yeah, safe, then so be it. And we're gonna open it. I also want to show you another one. This is a slide box, a very cheap slide box that I also once bought. Okay, this was actually, I don't know, this was one of the cheaper ones, and this one it's unfortunately totally unusable. I mean, on the outside, it looks kind of nice, okay? But look, if you take a slide, it won't fit. Can you believe this? A slide box where essentially it won't fit. I need to focus here. Yeah, and how is this possible? And the reason is because this side here, this wall over here, is bent inwards. So I think when they manufactured it, when the plastic cooled, it kind of deformed a little bit. And what you can do, and this is what I have done, is I've used a hot air dryer and I heated it up and I carefully bent it outwards again, and then it worked. Right? I mean, I shouldn't have to do that, right? That's one of the disadvantages of the very cheap ones. And another thing I don't like so much is a question of taste, really, but I don't like it if they actually glued the paper in here, right? Yeah, actually, I want to have a proper piece of paper or cardboard that you can take out and label outside and then you put it in. But I don't like that. But again, that's a little bit the problem of the very cheap ones. You're not even able to fit the slides in here, right? Because indeed it's bent here. And yeah, on here it's not even here it's possible, right? Not even on the edge here. Yeah, and some of the boxes actually it did work, and then I discovered that actually it's this the side over here that's kind of bent inwards, right? So that is essentially always the thing with those slide boxes. Yeah, if you have any comments or questions, please feel free. And you probably already know, some of you at least, that then I started because we've got now I designed my own boxes because I've got a laser cutter, so I bought those hinges and these things on Amazon. Yeah, so that is yeah, basically a homemade version. And yeah, here I put my own samples in here. These are basically sand slides and so on. So this is also a possibility that I've been experimenting around with, and yeah, a little bit simply because I liked to make my own things here. The reason why I put the logo so big over here, it's not only for branding purposes or for whatever, is this because then you actually know what the lid is. Otherwise, there's the danger that you open it the wrong way around and then all the slides fall out. So that's actually the reason why I put this strong logo on here, right? So, but that is a little bit the thing that I've been using up to this point. Side issue, I know that I remember that at the university times, many years ago when I was at the university, some of the students made their own slide boxes and they used corrugated cardboard. That's cardboard that has those wavy, it's called corrugated cardboard, and they kind of made their own slide boxes this way and used those cardboard, those wavy cardboard to actually hold the slide, which also worked quite well. Okay, so what I'm going to do now is I'm going to interrupt myself, and before we actually have a look at this here, and before I show you the slides that I have, I'm just going to quickly read the comments again. Okay. Okay. Jesse is 3D printing one. That's very nice. Okay. This is definitely true. I have a few antique slides I have bought that have been stored vertically, and the mounting medium has collected on one side. Thank you very much for this comment. I just heard about this, but I did not know if this is actually really a thing. Apparently it is. Yeah. Easy fix, put on coffee warmer for 5 minutes and the mounting media reliquefies and levels out, then store horizontally. Not sure if it will work for all antique media, but so far it worked on all. Thank you very much. Maybe if it's glycerin gelatin, because that can melt. Yeah. So that is a very valuable information. Thank you very much. Okay. I have that kind of slide box pre-made slide with 100 pre-made slides. Okay. Yeah, I arrange all my slide boxes vertically. That is another. That's another one. Yeah, if you if you actually keep that. Honestly, I didn't even think of that. If you put them in here, you just store them vertically. Gee, of course. Yeah. Yes. Yes. Yeah, sometimes the answers, the solutions are easy. Okay. Yeah. Yeah. Oliver, why don't you keep the slide boxes vertically? That's correct. Gee, I didn't think of that. Doesn't matter. It doesn't matter. I'm going to show you in any case this one over here. It's wrapped in plastic, so I have to open this. But this actually does have an advantage, storing them. The disadvantage is that it's kind of it takes up a lot of space. Okay, it does take a lot of space. I can't open it. It's difficult. Let me try. Here we go. Okay. So, okay, here we go. And this is the cardboard version. Okay. Yeah, made in China. Where else? But doesn't matter. Yeah, this is how it looks like, right? Made of purely of cardboard. It's fairly thick. It's around a centimeter thick. Huh? It could be a little bit flatter. It's slightly bent. And I think the reason why it's slightly bent is because it's been glued together. So maybe and then when you open it like this, then it looks like that, right? You see, even this is pretty thick. And yeah, I was quite generous here. It's actually quite nice. Right, and I'm going to now switch over and I'm going to show you, because I've filled 2 of them already with my antique slides. And if you want, we can also look at a few of the antique slides again. A couple of months ago, I already did look at some of them with you. So that is now the one with let's replace this here. Okay, and when you open it up, it looks basically like, it looks kind of nice. So it is indeed more easily visible. Right, and if you want to take them out, what you do is you just press in here and it pops up, right? Because there is a kind of it's a little bit elevated over here. So if you push down here, it kind of pops up, and then you can take it out more easily. So it kind of looks nice and it's easier to see a little bit. But again, I'm kind of worried if I drop the whole thing, then everything. I don't know. Yeah, so it would be kind of nice to have some possibility to kind of close it, right? I don't know if it's cardboard, it's paper, so if you spill something over it, we know, so wood would have been better, but I get it that it's cardboard is probably easier to make. And then my biggest worry was a little bit, what happens if there's kind of pressure on here, right? That's bad, right? But look at this. Then I realized the people who designed this were actually thinking a little bit, because look over here, the specimen part. Which first of all, it's fairly thick anyway already, right? So the cardboard goes above the slide. But look, basically there is this thing here. So the central part here actually does not touch the cardboard. Yeah. So there is actually no possibility to apply pressure directly on the cover glass because of this part here, which is further down. Right. So in that sense, just wanted to show that to you. And what I'm going to start making now is I'm going to probably think about making at least for those for those slides, maybe a separate wooden slide box where I can actually put several of them in here. Yeah. So that's the second one. Look, I like those paper wrapped slides as well. They look kind of nice. Yeah. So that's basically how this looks like. At least for now, I'm just gonna keep them in here. But actually, that's correct. I mean, why don't you just store the slide boxes like books? Obviously, yeah. it's probably even more space saving this way, right? Okay, didn't think of that. Yeah. So I'm going to read again a couple of your comments here, even if they were not directly. Why don't you? Let's have a look. I'm just gonna skip over. What type of microscope slides are you using? Well, what type? Well, I can quickly tell you a little bit what type I like to use. If this is already, I mean, I could actually answer those questions a little bit later as well. But what I do is I reuse the slides, and for this reason, I like to use slides that have basically polished edges, so that don't chip as well, and also angles here of 45 degrees, because I keep on washing them again, and then I reuse them, right? The company, again, this is not sponsored because they didn't ask me to do a sponsored thing, but they just sent it to me without preconditions. The company Globe Scientific sent me those slides here, and these were probably the highest quality slides I've ever had. They have a place here to directly label it, and I say highest quality because they were extremely clean when I took them out, right? They have also got edges that are polished and 45 degrees. Yeah. So yeah, just saying. Yeah. So ultimately, I would say there's not a huge difference between the slides. Yeah. So I'm going to quickly go scan ahead. I'm just reading if there's anything. Yeah. If there's anything relevant for me. Horizontal layout is in little trays is much nicer, so because you can see them all. That is indeed the case. Yeah, it looks a little bit nicer in that sense, but then again, yeah, one has to be, of course, always careful. It's not quite as it's a little bit bulky, maybe still. Yeah. So let's move on. There are no more comments. Yeah, out of curiosity, have the size of the slides always been standardized? Well, honestly, good question. If you the slide sizes, I think has been pretty much standardized. I think it's 3 inches by 1 inch, and for this reason, I think there's not a huge difference. Yeah. So if you just compare an old one and a new one, there's maybe a millimeter off or something, but pretty much the same size. Yeah. So it's 3 inches by 1 inch, and I guess. Maybe those numbers are a little bit arbitrary why they were chosen like that. Yeah. So that's basically the thing here. I don't know why not. Let's just look at a few of those. There are a couple of them here that I really like. Who cares? Let's just, what is this here? It's a cross section of a plant. Plants are always very nice. Let's just have a look here, just for those of you, so that we all do a little bit of at least a little bit of observation as well. Okay, we switch over. Yeah, let's move this away. And the arrow. Let's. I can always. And plants are always very nice to look at, and you see that those. Those plant cross sections, I mean, really, those old slides, really, those folks really invested a lot of time and energy and really to make high quality stuff. So that is again a couple of questions. What cover slips do you recommend, and how old are your antique slides? Okay, the cover slips, I don't recommend anything specific. I just tell you what I'm using most of the time are 18 millimeter cover slips, and I always, when I have cover slips, I always have to dry wipe them because they're always a little bit dirty. Yeah. I always use square ones. Occasionally, when I actually make permanent slides, like the ones over here that I have here, then I use round cover slips. I always have to change back. Yeah. So sometimes I use round ones. Maybe you remember the slide from last week. Yeah. I would say it's pretty much a question of taste. Yeah. Unless you want to ring them, then you have to use round ones. Yeah. But otherwise, Yeah, just for casual observation, it really doesn't matter a lot. Yeah. How old are they? Honestly, I had to do a little bit of research on some of them, because sometimes for the higher quality ones, the manufacturer is actually written on them, and then some of them were actually around 1900, so over 100 years old. And they were made, I would say, yeah, by companies, because at that time, especially in Great Britain, UK, yeah, in the Victorian microscope slides were quite popular. There were quite a lot of manufacturers there. In Germany, they were quite a bit into making microscopes. Yeah, Zeiss and some other companies, and in Great Britain they were pretty good also in making microscope slides. So a lot of there are lots of nice microscope slides available from there, and there is actually a database on Microscopy UK, and there is a small database, and I did a little research, and all of the historical microscope slide manufacturers were listed there, and that's how I was able to determine from what time to what time they actually made the slide. Yeah, and this is over 100 years old, some of them. Yeah, so this kind of is quite a nice thing. Let me always switch over to yeah. So for example, I just want to show this to you, what they have done. Yeah, so it's difficult to see here. So for example, W. Watson and Sons, London. Yeah. So this is basically many of these slides actually have a company name written on it. Yeah. Some slides of the antique slides actually have a specific the ring. If you look at this ring over here, right, you see that there are sometimes multiple colors in the ring. There was also some kind of a branding thing here, and here you've got the Arthur J. Doherty. Yeah, yeah, that's the company. Yeah, so. You see that, yeah. And if you just look at this slide here, I mean, look. I don't know if you're able to see this. It's way significantly thicker, right, than the modern ones that we have. Yeah, maybe a half or a third is thick. I don't know if you're able to see it. Huh? So this is quite a nice thing here, right? So just wanted to show this to you, and yeah. And some of the slides actually can be quite expensive as well. Yeah, but so I got specifically those slides that were a little bit more affordable, right? So let's read a few comments again, and then I would like to move on. How are your antique slides? If you store the slide box upright like a book, the slides lie flat. When sorting, just make sure the cover slip is facing backwards, or it will be on top later. Yes, okay. Okay, I have antique slides that are really tiny and others are super thick, but very nicely decorated. I believe it took some time to get some standardization. Yep. Okay. Yeah, Jesse ordered a whole bunch of stains here. Okay. So I go down here again and just skip. So when I tried PVA as a water-based medium, sand-like particles, no idea. So this is interesting. Yeah. No, sorry, cannot help here. Yeah, there seem to be some impurities in the PVA glue. Yeah. So maybe not everything was dissolved. Yeah. So so that's basically the short thing that I just wanted to share with you. Okay, because. Yeah, I always want to experiment around also with new things and new ways, and that's basically the newest thing that are those slide folders. So we're back here now to the desk, and I'm just going to move on now because I wanted to prepare a little bit of honey. Here it is. I've got and also some months ago, a big thank you. A microscopy friend from Germany sent me this honey over here. Yeah, look, we've already eaten it up. Yeah, and natural honey. So that's why it's labeled in German, because of course it's from Germany. This one over here was bought by a local from a local beekeeper. The lady who sold it to me on the fair, basically said that these are from different times of the year. Right. So, for example, this one over here is the early spring, and this is kind of later spring. And the honey is different. And when the honey is white like this, then this is because it started to crystallize. So these are actually sugar crystals, and this one here is a little bit more liquid. And I can actually see that it's a little bit white down here. So I wonder if this is some kind of some pollen and other particles that maybe have settled over the months. I don't know. Or whether it's already started to crystallize here. But in any case, what I already tried today is I tried to get the pollen out of the honey. Why not? And my idea was a little bit to kind of compare the different pollen that you find it from different plants. I did not find a lot of pollen, right? But there were a handful, and I just thought that I'm just going to show it to you what I've done, and yeah, let's have a look, okay? So yeah, so what you need is the following. You need, of course, a centrifuge. I've got one over here. Yeah, this one, it was really a super cheap one. Let me put the plastic away. Yeah, so it was a fairly cheap one, but there are also hand centrifuges, and for me, this one has been doing its job, right? It's a low-speed centrifuge, around 4,500 rotations per minute maximum. Yeah, you probably don't want to leave it unattended because I don't know how good the quality is. If yeah, but it's been doing its job. And another thing that you need is you need some yeah, these are not test. I need to switch over. These are not test tubes, but these are centrifugation tubes, right? I also use them to collect water samples or other samples, so I also take them along. Yeah, every time when I find something of interest, I either put them into those, or you already know, of course, this is something that you already know from my previous videos. These are much larger centrifugation tubes, which are also used to keep samples. But my centrifuge will only accept those here. Yeah? And that's all that you need, right? So what I'm going to do now is the following. I'm going to add a little bit of honey into the tubes. I'm going to add water, and I'm going to dissolve it, and yeah, 1 tube per honey, and then I'm going to spin it down using a centrifuge, and I'm going to then put the pellet under the microscope. That's it, right? So I actually thought about preparing everything already for you, but then I said, no, the point of a live stream actually is that you kind of follow along a little bit also in real time. So yeah, and also see some of the problems and imperfections that I'm doing. So this one over here is that's the early springtime honey. Let's move this up a little bit. Yeah, and I use a fresh white toothpick. Yeah, for the lack of a better tool, maybe. Yeah, and I'm just going to take out some of this honey here. And we're going to put it in here. And the more, of course, the larger the pellet. So I will just estimate from the amount that I had today in the morning. Because usually all of the things that I show you, some of the things are improvised. But if I try out something like this, of course, I want to make a little bit sure that actually there is something to see. So I usually try it out. Yeah. I think this is already more than enough. Okay. So that's number 1. So I'm going to leave that here. I'm going to close that. The toothpick I'm just going to throw away. And this was actually quite easy to take out because it was pretty solid. But this honey over here is a little more difficult. Today, by the way, for your info, today in the morning when I tried this, I also tried using a pipette to take up the honey, but it didn't work well because it was so thick that it would barely want it to go into the pipette. Yeah. So, and that's another one. Put this away here. Yeah, here this one is already see a little bit more difficult because it's much more liquid. Okay. So here we go. It's kind of surprising how much the consistency is very different. So maybe this is actually enough already. I think this is more than enough. Get another one here. So, and now what we do now is we have to add a little bit of water. Okay, so let's put this away because I want to make some space. Yeah. If you heat the honey, would it make? Yes, definitely. If I heat it up, then it's going to be much more liquid. But I found out that actually, yeah, the way that I've done it here is fine. Yeah, it's heating it up would probably in a water bath. It would probably take way more time. So and then I usually that's approximately the amount, and then I usually fill it up to around 10 mils with tap water. Okay, some people ask me, should you not use distilled water? And I said, no, actually, it really doesn't make a huge difference because the impurities in tap water, yeah, are nothing compared to the impurities that we have when we actually mix it with honey. Yeah, so it's not going to be distilled anyway. Yeah, so also approximately up to 10 mils here. And now we've got to be patient. Okay, we got to be patient now because I have to now dissolve this. So I put on a cap here. Okay, and now we have to be super patient. And this is actually where maybe warm water would be better, right? Yeah, so it's... Oh my gosh, this reminds me of something. Back in the lab, back in the day when I was still at university, long time ago. I did my master's thesis in bacteriology. It was called systematic bacteriology, bacterial systematics. And this basically means is I did biochemical analyses of bacteria to find out what they are. Of course, nowadays you use a lot of DNA analysis. I also did that. But what I did is I extracted fatty acids from bacteria. And then I put them into so-called a device called a gas chromatograph. And then the fatty acids were analyzed, and then basically the results were then interpreted, and then you kind of could estimate which bacteria you were talking about, which you had, right? And I remember that I ended up spending hours shaking test tubes like this. Yeah, because they had to be carefully extracted, those fatty acids. And it was actually, yeah. I remember I was going both hands, 5 test tubes in each hand, yeah, and yeah, kind of shaking them, yeah. Nowadays, of course, why don't you just use a machine? But the thing is that those mixing machines, they were too aggressive, and they had to be gently extracted, and so basically by inverting the test tubes, yeah. So that is kind of what it reminds me a little bit, yeah. Do you know botulism? Yeah, so that's kind of the worry that essentially there could be the bacteria Clostridium botulinum in honey. That's the reason why fresh honey is sometimes not recommended for infants, for babies, because they could get a bacterial infection. Yeah. I don't know to what extent this is really an issue or not, but I have heard of this as well, right? So yeah, you see over here the honey is slowly starting to dissolve here. Yeah. You have to be especially careful that there is nothing here on the bottom. Yeah. So this is already completely dissolved. Yeah, so we can put this away, but over here there's still some honey, I think, sticking over here. Let's do it like this. There's some foam. The foam doesn't really matter a lot because, yeah. But I want, if I really want to make sure that all of the pollen are properly extracted, I have to make sure that there is no honey left over. But you know what? I'm going to do it now the easy way, because there's the honey sticking here on the surface of the tube. Let's do it like this. And then I have to tell you something that I discovered today about these test tubes, which kind of I didn't know. But yeah, I just want to share with you as well. It's something that I was a little bit surprised by, and it's difficult to see. I've got 2 different test tubes, and I don't know if I'm able to see this here. But you see over here the how do you say, the lines, right? And then in one of those test tubes, I think it might be this one here, the lines, they continue on the inside of the test tube, but it's not over paint, but it's the plastic that has lines in it on the inside. Yeah, it's impossible to see now. Yeah, and you have to be very careful that those. So basically, it's not smooth. The inside surface is not smooth because it kind of the lines kind of continue. And this basically means that the pellet got stuck not down here on the bottom, but here where those the lines are on the inside. Huh? So you have to make sure that when you insert the test tubes, make sure that those the lines here are not pointing outwards but inwards, so that the pellet is able to collect it all the way on the bottom. Yeah, just saying. Huh? Yeah. So the next thing that you want to do that's yeah, and some of the community members have got the same centrifuge. And it says centrifuge, and they said it doesn't really matter because it's not really of high speed. But you should more or less make sure that both of them have the same how do you call this, the same mass. Okay, otherwise there's too much vibration. So this one here is about 1.1 grams to light. I mean, some people said that it really doesn't matter a lot. Yeah, I just want to play it safe and get it more or less correct. Yeah, I think that's gonna be okay, right? So so that's basically it, and we can now spin it for maybe about 5 minutes or so. It's gonna be a little bit noisy. Okay, so I don't know if, it's going to be a little bit noisy. So then let's have the centrifuge view. Lid goes away. I will insert it now, not with the lines outwards. I don't know if you're able to see it like this, not with the lines outwards, because remember there might be those on the inside, those tiny plastic lines where the pellet and the solid material gets stuck, right? So I don't want that, so I'm going to turn it around like this. Yeah, that's one thing. And over here, I'm going to do the same thing over here. Just want to make sure that not the lines are not on the outside, but the other part is on the outside. Yeah, and the lid goes on top, and then I can actually turn it up. Yeah, there is actually also a timer function, but I really. Yeah, maybe you can hear the clock. Yeah. Let's spin it up. I don't know if you're still. The big jar. I have got it over there. Okay. So Tini is asking the mixed. I don't know if you're able to hear me. I wonder how your big jar of mixed samples is from last week. I wonder how the introduction of other sample affected it. I will tell you the following. I did not check yet. The only thing that I realized is that the water fleas are all gone, unfortunately, right? But the algae are still there. Some duckweed is still floating around. Yeah, there is still stuff there, and if we want, we can put it also under the microscope. Yeah. So, but let me quickly check again if there's any comments. Yes, Oleg is asking me, what have you... again, these are off-topic questions that I actually wanted to save for later on, so that I have something later on to talk about, right? Yes, I use 16S, not rRNA, but rDNA, ribosomal DNA. The 16S RNA, we didn't sequence, but we sequenced the DNA that codes for the 16S RNA. So it's basically the same sequence, right? And then that's what we've done. We simply got an email with a base sequence, and then I did an online database search, and that's how we, So I would say that's the gold standard, the 16S rDNA, ribosomal DNA sequencing. It's pretty cool, I think. It's fairly easy to do, not so time-consuming to do, and is very powerful. So all of this biochemical analysis that I have done is sure, it's interesting. I learned a lot, but I would say that probably the 16SR sequencing is way way more efficient. Yeah. I can tell you stories about this. We're just gonna wait a couple of more minutes. So that is. So I'm now basically at the maximum speed. Yeah. Yeah. 500, 1,500 p.m. for 5 minutes. Yeah. I'll just give it. I just give it a couple of more minutes. Yeah. What else is there? Yeah. Yeah, I'm just reading in case you're wondering. I'm just reading your comments. Okay. Top chat. Maybe I should go live chat. Maybe then the sequence is a bit different. Yeah. If you go live chat, then the sequence is a bit different. Water flea. Okay. Water fleas. Yeah, the water fleas are all gone. That's yeah. Maybe I didn't feed them enough. Yeah. You found an annelid. Paul said he put some algae out and then he found an annelid. I suppose, I don't know, I'm going to guess it's probably Aelosoma, the oil droplet worm, those cute worms with a big head and the red dots, if that is the one. Is it a problem for my aquarium? No. I mean, they're normal part of the, of a healthy ecosystem. If you've got an aquarium, I guess fish are also going to eat those. Yeah. So I don't see a problem with those. You know what? We're just gonna. I'm just gonna say that maybe. Yeah, exactly. Jesse is saying, yeah, often the fish will just eat them. So and now comes the interesting part. Now I need some yeah. Let's take it out carefully. Is there a pellet visible? Tiny one. Okay, tiny one. What about this one over here? This is a little bit better. Okay. So, I'm going to quickly go back to the desk view, desk view. So I'm going to show you now what we've got here. Yeah, see, it's much more clear right now. And you're probably going to see this tiny little, I don't know, shall we use a dark background? It's a little bit difficult. You see this tiny little thing down here? That's the pellet. And that's what we actually want to have, right? So, and what I do now is, if you're brave, you can just dump it out. I say if you're brave because the pellet has to be sufficiently stable. Yeah, and I've already tried it out today in the morning, and that's why I know that the pellet is sufficiently compact. It's not always like that. Here, as well. The pellet is over here. It's very tiny. Yeah. And that's it. So there is now a drop of liquid left here. A tiny drop of liquid. And what I'm going to do now is I'm going to just resuspend it. And in order to do that, I'm just going to use my microliter pipette simply out of convenience. Because it's easier for me. And then what you do is whatever is the tiny, let's go down here a little bit. Now you can see the pellet a little bit better, right? And there's now a little bit of liquid left. I bumped camera. And what you do then is you simply carefully resuspend it by drawing it in and out. And that's it. You take a small sample. You take a microscope slide, hopefully a clean one. That's a little too dirty. Why is it dirty? Because I washed them. And I didn't wash them properly, I guess. Okay. And it's okay. Let's put it on here. Now I forgot which one I took. I think, no, that was not this one. This was the one. Yeah, that's the one. So let's put it away. So, and a cover glass. Cover glass. Yeah, it's a bit dirty again. Need to refocus. Yeah. No red dots. Okay, so then maybe it's not a iolosoma. Yeah, and here we go. Bloop, and let's go into the microscope view and let's have a look if we're able to see something. I'm quite sure, of course, we're able to see something because we should be able to see something. And here we go. And we're going to go way up with the magnification. Lots of particles. I mean, I remember a couple of years ago when I've done this as well, I found also some parts of some bees. Yeah, maybe we want to go up with the magnification further. Yeah, lots of solid stuff. Yeah, and this round thing that you see here right in the center, that's already a pollen grain. So, and honestly, I am not so much into pollen identification. What you then, and apparently it's not so easy either. But then what you'd probably do is you take some kind of a manual and then you kind of check a little bit what pollen it could be, right? So actually folks who are keeping bees, they're doing that. Yeah, so that's if you want to have a little bit of color. So with color, so that's now 60 times. And one of the things that I wanted to do this spring and I didn't is actually to observe pollen grain germination. So what you do is you collect some pollen from flowers and you add a little bit of sugar water, right concentration. Then if you're lucky, you're usually able to see that the pollen grains start to germinate and form pollen tubes. You see lots of other stuff is in here as well. Probably also some bacteria. Here's another pollen grain, right? So you see that honey being a natural product is quite a complex mixture of a lot of different things here, right? Yeah. I'm seeing here's another pollen grain. Yeah. I remember a student of mine a couple of years ago, 3, 4, 5 years ago, actually did a study of different types of honey and the pollen that you could find as a science project. Yeah. Here's another one. Yeah, and all of these things that you see floating around in the background, I don't know, it could be anything. I mean, maybe bacteria, of course, yeah. But because honey contains so much sugar, therefore there is, it doesn't spoil so easily. Yeah? Because there are also natural disinfectants in there. But I did also have sometimes some honey that maybe microplastics, yeah, wouldn't surprise me. Honestly, you find those, yeah. But honey apparently is also filtered, yeah. So I can imagine that any larger particles are also removed. Yeah. Again. So you see, there is so if you're interested in stuff like this in pollen identification, I mean that's what actually some folks are doing. Beekeepers, they're actually assessing the honey quality based also on the pollen that they find. Yeah. Could we stain the pollen to see them? Well, I will be honest with you. The pollen themselves don't have to sperm, but they will actually form when the pollen tube starts to grow. So what you actually have to do is you have to cause the pollen tube to grow, and then the cell is going to start dividing, and then you have actually 2 sperms. So that is it's correct that there are 2 of them but they're not present from the very beginning. Yeah, they will form as the pollen tube starts to form. I don't know what this is. Sometimes you cannot automatically assume that if you find something weird in a sample, you cannot automatically assume that it's in the sample itself. It could be something that was some kind of dust or dirt on the slide itself as well. So only if you actually find it at sufficiently high concentrations, only then you can actually say that that it's actually really part of the sample. Yeah. If you leave a bit of honey out on the slide in the air, could we see the sugar crystals too? We're going to try this out. Thank you. I'm going to put the crystallized sugar then under the microscope, right? Directly. And then maybe we're also able to see the sugar crystals. That's something I did not try out yet today. But you get the idea a little bit. You know what I'm going to try? Let's just go phase contrast, because this is always something I like to do, just to get a different image as well. Yeah. And very often, you can actually see now any bacteria that are present can be seen as dark dots. Yeah. So if you, I mean, there are lots of dark dots. I'm not saying that all of them are bacteria, right? But it could indeed be that some of them are. Wouldn't be surprising. Is also not a problem. Yeah. It's just a normal part of the honey, right? But that's phase contrast now, and phase contrast, as you might already know or not know, is pretty good at making very difficult, transparent cells better visible, like bacteria, for example. Yeah. So so what I'm going to do now is I'm just going to try the second one as well. I'm occasionally also checking the time a little bit. So let's try the second one as well. I need to get myself another fresh slide. Checking it if it's reasonably clean. It's almost reasonably clean. So here it is. A new pipette. And that is now, okay, that is now from, the one that we looked right now was the earlier one. And this one is now from the forest. It says here, what does it say here? Wald und Blütenhonig. So that's basically forest and flower honey. So basically from forest flowers. Yeah. So we resuspend. We resuspend the yeah. And let's give it another try here. Yeah. Okay, small amount. And I mean, I tried it today, of course, and it looks very similar. So don't expect a huge difference now. Yeah. But the idea would be, if you really want to do this a little bit on a more serious approach, to probably compare the different types of pollen that you find. That's way out of the scope of what we're able to do now. And the study of pollen is called palynology, and it's a science. It's yeah. I'm still now in phase contrast. Let's get a better overview. Okay, that's basically in bright field. Okay, here it is. No, this no, it's again DIC. Yeah. Yeah, let's see. Okay, here there might be again those round things here. Let's go in a little bit further. Yeah, these are again pollen grains of different types. But otherwise, it does look quite similar, right? You get the idea. Yeah. So it's also maybe a way, because I know that there are also artificial honey that is being sold. Yeah, and natural honey, however, contains all of these other things as well. Yeah. Yeah. So let me have another look here. Yeah, lots of stuff here. Yeah. It would be cool to simulate plant reproduction using ovary and pollen and try to replicate it. Okay. Yeah. I mean, that is. Let's put it this way. That's a little bit difficult to sometimes see biological processes like these, which are so structure dependent. What they usually have done in the past is you cannot actually see it live, but they made cross sections and staining of at different time intervals, and that's how they were able to determine what's actually going on. Yeah? But actually observing a process live is sometimes difficult. But look at this thing here in the center now. Let's go up. Let me see. Yeah, these look very much like rod-shaped bacteria. Those long things that are kind of floating around. Could be rod-shaped bacteria. Again, this doesn't mean that the honey is bad, right? That's just a normal part. Yeah. But I do get the impression a little bit that there seem to be fewer pollen in here than in the other one. Yeah. Indeed. I've only found a couple. Yeah. Oh, what's this long thing here? I don't know. So, one of the interesting things is I have also put honey under the microscope that contained here is also something here, honey under the microscope that actually contains some hair of some of the bees. Yeah, I've not seen anything yet here. So. Yeah. So you know what I'm gonna do now? What time is it? It's 55 minutes in already. You know what? Because there was the question. Here's another pollen. There was the question of maybe putting directly a specimen of the crystallized honey under the microscope to have a look how that looks like. Yeah, we're gonna do that as well. Because why not? Why not? Where is the desk? Here we go. Yeah, maybe I need some tissue paper because I don't like it to have water. Okay. So, it's always a good idea to use a fresh and clean object when you're actually picking out something from honey. You don't want to contaminate it, obviously. And all you need is a very small amount. So, and this is way too much already. So let's just put it on here like this. So, and in this case, we want to watch the see sugar crystals if present. I'm going to quickly wipe the cover glass as well. We put that on top, and we press down. We press down until we get a very thin layer. And yeah, so this is going to be a little bit of an interesting thing now, also for me, because I've not done this yet. So scope view, it's way too bright. Let's take it out. And now this is the crystallized honey. Ooh, it actually does look kind of nice. Okay. This is now basically bright field, right? No polarized. Yeah. And with polarization, looks kind of nice, I guess. A little bit like modern art. Let's go directly to the edge of it. Yeah, here that's the edge. Let's see if we can change colors. Oh, don't you like playing around like this? Yeah. So this is that's basically these are the sugar crystals. Yeah, here are some questions again. Thank you very much. Many of the questions arrived. Many of them are already answered. It doesn't matter. I'm just going to put them over here. In a couple of minutes, I will respond to those. Yeah. So let's go up yet further. Actually, it does look kind of nice. And so that is honey under the microscope, sugar crystals. Yeah. Will it crystallize better without the cover glass? Could be. Let's put it this way. I already put crystallized honey on here. But when you use it without a cover glass, I can imagine it will crystallize because it will lose water. The water will evaporate, and therefore it could be that it starts to crystallize faster. Yeah. So I kind of like this actually. Yeah. Again, this is because of polarized light. So you need polarization filter, and because of my DIC setup, I've got polarized light already. Let's go up yet further with the magnification, simply because, the depth of field really goes down now. Yeah, but you can play around with the colors like this. Yeah. So if you just joined, this is honey under the microscope, and of course we're now going to try something. And you know what I'm gonna do. I'm gonna add a little bit of water. Let's see if we're able at low magnification, because I don't want to get the objective wet. That's also an important thing here. So this is now at low magnification. Okay, let me see. Here we're basically at the side, right? So, and what I'm gonna do now is I'm going to just bright field. If you want, this is bright field. Okay. So this here is bright field, okay, 10 times. Phase contrast I did not try yet. So let's try phase contrast just for comparison. Need to open the That's phase contrast. Actually, also looks kind of nice. It's more black-white. Yeah. So that is, look, it's still a little bit liquid. It's still flowing a little bit. Okay. So for those of you just joined in, I just put some honey under the microscope. These are the sugar crystals. Yeah, and sugar crystals of honey. Let's just stay like this. And what I'm going to do now is I'm just going to add a little bit of water. Because, why not? Because maybe we're able to see some of the sugar crystals to dissolve. And then I'm going to maybe talk again about a couple of your questions that you've got. So, I need a little glass beaker here where I'm going to add a little bit of water. Yeah. I need to tell you something else a little bit. Do you actually see the stage with the slide on the top? It's black-white now. Yeah. The reason is if there's not enough light present, it will switch to black-white. But sometimes if you do something like this, yeah, if you go with the then now we're back in color. Okay, it's a little bit of a yeah. So I'm going to now add some water, and let's have a look what happens to the crystals here. Okay, you see how now we can go up with the magnification a bit. Do you see how the water starts to dissolve the sugar? It looks quite cool. Let's change colors a little bit again. The water starts to stream inwards. Yeah. Look again. Let's go here. Yeah. Now we're in color again. Yeah. I think it's quite nice. And what we can then, I don't know, I could leave it as it is, but then usually when the water starts to evaporate again, then sometimes you're able to see hopefully recrystallization. I don't know, sugar does not recrystallize quite as quickly compared to salt. Yeah. This is actually quite interesting. You see how the water starts to stream inwards, and as it goes along, it kind of dissolves the sugar. Yeah. Of the honey. Yeah, it looks quite cool. Yeah. By the way, I like doing these things. Yeah. In case you have not seen it yet, today I uploaded a YouTube short, a very short YouTube short, about dissolving salt and recrystallizing salt. It looks also quite nice. It's only a 20-second YouTube short. It's published in the same channel here. If you want, you can have a look at it. Yeah, yeah. Yeah, Paul says, I managed to get a picture of that very active worm. I posted it in the forum. Okay, thank you. There is debate about whether honey is actually honey, right? And not like a huge percentage made of sugar syrup from sugar. Yeah, I mean, there is artificial honey out there. Yeah, so apparently it's not even that. I know that even my grandparents told me that long time ago. I mean, honey was expensive, so they actually made honey themselves artificially. Yeah, and just by adding a lot of sugar syrup and then adding some different herbs or taste to give it some taste. So that's then artificial honey. But I mean the natural honey, you can see that essentially there are a lot of impurities like pollen in it as well. You don't see them here so much because this is not concentrated, right? I was actually using a centrifuge to concentrate it, and of course also if there are pollen grains visible in here, okay, they are covered up by the sugar crystals. Yeah. Yeah. Couldn't we try to use a blow dryer to dry out the slide faster? Yes, or use the coffee warmer. So that would be another thing. Yeah. But you know what? I will take this as an, how do you say, a suggestion for the next video, next week, where maybe we're going to do a little bit of recrystallization, but not of. I mean, I've been doing a lot of salt crystallization and vitamin C and so on this channel, but actually not of sugar from honey. So And so there are a whole bunch of other quite interesting crystals out there, like aspirin, salicylic acid makes nice crystals, caffeine makes nice crystals. Yeah, yeah. And here we go. Here we have, of course, the sugar from the honey, right? So it would be interesting to study the two honeys from one flower and one. Yep, yeah. That's yeah, yeah. So that is kind of how it looks like. Okay. And now it kind of stopped, and I guess that it's going to start to slowly dry again and then recrystallize again. Okay. So you see everything here. These are bubbles. Otherwise, it looks quite nice. Okay. So what I'm going to do now for the next, I don't know, 20 minutes or so, I'm first going to clean up a little bit here. I've got a pipette and all of these things have to be gone. Yeah, short. What's inside a grain of pollen? DNA, cellular material, obviously. And the nucleus will start to divide, and then it will be able to fertilize the ovary, at least the ovum, the ovules rather, in the ovary of a flower. So that's what's inside a grain of pollen. Gee, cellulose, I would say, for the cell wall, and then other cell components. But I do admit that a pollen grain is fairly large as well. Yeah, so I wonder what is in there as well. Maybe some storage substances used as an energy source. I'm guessing now. So what I'm going to do now is I'm going to have a look at some of your questions again. Yeah, by the way, a short announcement. Yeah, many of you, and I posted this also on the forum, also many of you probably have already seen that my website and the forum were down, completely down for several days. Not reachable. The situation was so bad that even I sometimes could not log in to do maintenance work. The microscopy forum, by the way, it's pretty active, right, and pretty important for. I myself am not getting very active in the forum. I'm just a bystander a little bit. I leave it up to the community. But the forum and the website could not be reached. This already happened last August, but it's never been as bad as this time. So I emailed the support of my web hosting company. And indeed, within a day or so, I got a response. They fixed it, and they also told me the reason. The reason why the website was down is because there were too many requests, bots, automatic requests, from certain IP addresses, and they blocked those. So, and then somebody also notified me that there were over 3,000 guest users in the forum. All of them were bots. So apparently, maybe they were doing some AI scraping or reading or harvesting. I don't know. In any case, there were too many requests to the web, and this kind of brought the web page down. Right. So I just wanted to tell you a little bit in case you were always also a little bit surprised why you could not access the website or the forum. It is because indeed the site was under attack, and I had mixed feelings. I was a little bit happy that it was not my fault because I did not change anything on the website, but it was actually an external thing. Yeah. So DDoS protection, yes. It was. I don't know if it was actually a DDoS attack. A distributed denial of service attack, or whether it was actually some kind of AI bot scraping the contents of, I don't know. So that's essentially the how do you say the issue. So the web hosting company blocked those IP addresses, and now hopefully it's gonna work again. And they also recommended that I install some WordPress plugins that kind of protect the website and so on. So just saying in case you wondered. That's number one. Number two, this kind of made me realize a little bit something very uncomfortable, how extremely dependent we are all on external factors. I mean, if YouTube closes down, which I don't think is ever going to happen, hopefully, but then what happens to the community, right? So over the last couple of years, what I've been doing is I've been collecting newsletter email addresses over the website, but I have to admit I have not sent out a lot of newsletters, any newsletters yet, because I just prefer doing videos. But what I'm decided to do now is maybe at least once a month to yeah, maybe send out a newsletter with some microscopy tips and maybe some questions answered. Yeah, so that is simply something that I yeah, will try to do a little bit more, because in case something happens to all those websites, that at least we have some kind of an email possibility. I also have a social media site on Blue Sky. That's also something that I recently started. The reason why I use Blue Sky is also because you can actually read the posts and the pictures even without an account. Right, and that's a certain advantage as well. Yeah, so Jesse is saying it will probably, unfortunately, probably come back, but fingers crossed it doesn't. DDoS protection will also protect against bots due to the way they act are similar. Yeah, so this is also a little bit my worry, because when it happened in August that the website went down, it disappeared again on its own. Then basically it was kind of okay for a couple of months, and then last week or so it was pretty bad. It was completely down. It was a disaster. Yeah. So we hope for the best. So what I'm going to do is I've got two pages here. Some of them are already answered. Okay, but that's fine. Okay, and some of them were already answered by other community members. Please explain the difference between paramecium and ciliates. In short, a paramecium is a ciliate. Let me just make some drawings. Okay, let's just make it. I don't know. Do I have a proper pen? Now I spilled my toothpicks all over the place. Yeah, if you look at the protozoans, the single-celled organisms, I know I'm probably simplifying this. You've got those. Let's just. Yeah, it's a single cell, and you have those that have tiny little hair all over the place. These are called cilia, and these are called ciliates. Yeah, so that's yeah, very common. Huh? Some of them look like Stentor, for example. Also it looks like a trumpet animal, and there has tiny little cilia over here. Made a video as well. And then you have got the so-called the flagellates. They are cells, and they have a flagellum. Yeah, yeah, it's yeah, either one or two of them. Yeah, and you have also got amoeba, which are able to change the shape. Yeah, so there are different ones, and paramecium simply is one of those classic model organisms among the ciliates. Also, probably because it's so common. Yeah. Okay. Oh, here's a news. Thanks for the recent newsletter you sent, Mail. Good initiative. Yeah. Okay. Yes, I've been honestly the newsletter. This has been a big, how do you say, not headache, stomach ache for me rather, because I started collecting the email addresses a couple of years ago, as I already mentioned, but I was very reluctant sending out newsletters because I didn't know what should I write, and is it, I don't want to spam people and so on. So there's a little bit of a format issue, and so I thought maybe once a month I'm going to send out some hopefully with valuable content, maybe with some questions answered or additional information on some of the videos or so. Yeah, so that's a little bit the idea. Yeah, and just saying. Yeah, as soon as I finish my 3D printed case design, I will be sure to send you a link. Thank you very much. If you send me the link to your 3D printed case, I'm going to print it out and I'm going to present it here also in the live stream. Yeah, yeah. Slide boxes vertically. We talked about that. That's a good idea. Yeah, vertically stacked. Didn't think of that. What type of microscope slides are you using? Honestly, yeah, there is not a specific kind really. I simply I'm choosing those that are a little bit of slightly higher quality because I keep on washing them, and the cheaper microscope slides that they usually have very sharp corners, and then when you wash them there is a slightly higher risk that you hurt yourself. That's why I was actually using those with polished edges. Yeah, amoeba flagellates. Amoeba flagellates. Yes. Yeah. Also these are also those organisms that for example, like slime molds and so on. Yeah. artificial pollen has ever been made. I don't think so. I made a checklist with an agenda, choosing tasks for daytime using Visual Studio. Okay, items from the digital system. This is a very computer programming related point here. Yeah. How old are these antique slides? Maybe 100 years old, around, at least according to my research, right? Simply by the company, age of the company. Horizontal layout of the trays is much nicer, also because you can see them all. Yes. Why the producer of the cardboard slide did not change the index in front? Then when you open one slide, you see the correct specimen. This is actually one of the criticisms that I also have, honestly, and I actually also, because I've already made an intro video of this that I'm going to post separately. Look at this here. I mean, that's a wasted opportunity, really. Look, it says here 1 to 10, right? Wouldn't it be kind of nice? You open it up, and then you basically have it right next to it, the 1 to 10, or at least in here. Yeah, but right now it's not really, how do you say, matched? Yeah, either in here maybe, yeah, or at least here. Yeah, I think honestly this would be much nicer. Yeah. Then again, you can always glue something over it. Yeah, maybe some piece of paper. But that is, I think that's a little bit of a lost opportunity. Yeah, actually, I thought about this myself. Yeah. So let's put this away again. Yeah. So, out of curiosity, have the sizes of always been standardized? I don't know about that, but I assume 1 inch by 3 inches. So, and that's why I guess it just stuck to that. Yeah. No specific recommendation. I get the cheap ones, the 18 millimeter ones. I also have very large cover glasses, which I rarely use. Depends really. Round cover glasses I only use for making permanently mounted slides. Yeah. So if you store the slide box upright, like in the book, yes, we talked about that. I have antique slides that are really tiny and others are super thick. I believe it took some time to get some of the standardization there. That could be. 100 milliliters of methylene blue, a whole bunch of stains. Yeah. When I tried PVA as a water-based medium, I noticed that there were some sand-like particles. I don't know about that. Were these some kind of impurities? Sorry. Yeah, I mean, the PVA glue that I'm using is completely transparent. If there's anything in there, then it should be a little bit cloudy, right? But yeah, I'm using completely. I know there is also white PVA glue, which is not suitable at all. I've got a case of 100 histology slides with the media being in varying states of cracked and dried out. Okay. Yeah, so this is actually, depends a little bit on how well the slides have been made. Yeah? To display for viewing in an attractive way. Well, generally, I don't know if this is a big issue, but maybe you want to keep them dark, the slides, especially antique slides, stored in a dark place, because, I mean, if they're exposed to the microscope light, it's not dark anyway. Yeah, but maybe sunlight is not such a good idea because UV light likes to bleach stains. Yeah. What's your method for washing slides? Okay, that's actually a nice one. Thank you very much for your donation. Thank you. Washing slides is essentially I just rinse them with hot water. So what I do is the following: I pour boiling hot water over them, simply to kill off everything that might be on there. And then I simply rinse them, and in most cases, you do not even need soap. I tried dishwashing, putting them into a dishwasher. This also works. Or simply adding a little bit of dishwashing powder in a container. So if I just show you over here. What I do is here, this is where my slides go. I pour hot water over it, and then I simply take out the slides individually, and I use my fingers to clean them. Right, you want to dry blot them as well, otherwise you get water stains. Yeah, so I tried all sorts of things, just hot water, without soap, dishwashing, everything. Yeah, why the producers of the cardboard slide did not change the index? Yes, that's what we talked about. Have the sizes of the slides always? Okay, that's the same. It's the same text here. Okay. Okay, or did I not? How old? I think that's there's a little bit of repetition here, but it's fine. Yeah. Sun-sized particles. If you heat the honey, yes, warming it up definitely makes it more liquid. 16S, yes, I talked about this. I did 16S rDNA, ribosomal DNA sequencing. Did you write which honey was in which tube? Yes, I actually labeled here. BL is that's the German abbreviation. It's flower. Blüte is basically flower, and this one is forest. Yeah. Yes. Now I'm actually labeling the things here. Yeah. So I got that. I wonder how your big jar. Yeah. What time is it now? The big jar is standing over there, and it's looking ugly. That's all I can say from last week. Water fleas are gone. Yeah. Some algae out of the aquarium, and there was an annelid. We talked about the worm. No red spots. Could be something else. Yeah. Humans, 99.9% of the nematodes are harmless. But then hookworms, yeah, you see, it's always like this. It depends also sometimes of the reproductive cycle, yeah, and how if you're eating contaminated food and so on, yeah, that's actually the issue. Yeah. That pollen have two sperm. I talked about that. Those sperm will form during the formation of the pollen tube. Okay. Yes, the sugar crystals would form, especially also when you leave honey out because the water evaporates, and this, of course, increases crystallization. Yeah, and it would be cool to simulate plant reproduction using the ovary. I don't know if you're able to do it. No, not under the microscope, so easily possible. Yeah, but essentially what they have done is they've made cross sections of plant tissue, and then they were actually able to figure out what happens when. Yeah. So let me have a look. Thank you again for the donation. Yeah. Yeah, I've been just recently checking again for some nice slides, antique slides, but I couldn't find at least any of those that at least I wanted to have. So there is I have a plastic sheet in front so you can slide info labels if you change the slides. Aha, plastic sheet. Okay. That's interesting. My little cat has intestinal problems. Can I use my microscope to spot parasites or eggs? Is it feasible? Honestly, my opinion, my general opinion about doing diagnostics, specifically on humans and also on animals, no, you want to go to the vet, period. You just go to the vet, no questions asked, it's your cat. And maybe you are able to spot some parasites, but maybe not. It depends on the content. You don't. It's too important, the health of your animal. So yeah, and it's also I probably wouldn't put certain samples under the microscope simply out of hygienic reasons. Honestly, right? So that's also another thing why I would be not do that. Yeah. Is it possible and feasible? Not really helpful. Yes. Yeah. If the symptoms get worse, I'd take the kitty to the emergency. Definitely. Yeah. Do you know which cover slips are better at uniform thickness for best photography? Not that I know, but I would not say that the uniform thickness is such of an issue. Well, first of all, I'm not saying that the thickness is not an issue of the cover glasses. Yeah, as a matter of fact, cover glass thickness is important. However, I think that any change in thickness that the cover glass has within the cover glass, maybe it's like a little wedge or so, probably is not going to make a huge effect, considering the fact that if you put a cover glass on water, that there is already some irregularity there, because a cover glass is also not flat and might also bend slightly, is also a little bit flexible, and there's capillary action, and then there are maybe some particles beneath it. So I would probably say maybe the actual cover glass thickness probably plays a minor role compared to the other factors that play a role. Yeah. So Yeah. However, having said that, I know that there are some highly specific applications where you really need to have a very precise, you're really going to the limits of the resolution of the microscope. And I know I've heard that some people, therefore, there are microscope slides that are, not slides, but cover glasses that are quite expensive, that are manufactured to a very high precision. So they're all the same thickness. And then I read somewhere that some people don't want to spend so much money, so they're using a caliper to actually measure out the thickness of the microscope slides to make sure that they're all standard same thickness. But they only do that if for specific applications, right? So you see, it's not a yes or no answer that I'm giving here. I'm just saying that it depends, I guess, a little bit on if it really has such a big effect for what you want to take a picture of, and whether there are not other factors that also play a role, right? But having said that, yes, people actually do measure out the cover glasses with calipers. Yeah. Yeah. So, hidden world. I'm reading again here. Do you remember the old photo album where you slide the photo in a sleeve? That system I'd use to index the slides in my box because you can slide out the label and put another name in. Are you talking about kind of like where you a system where you slide in the slides like they used to slide in pictures maybe? Okay. A couple of streams ago, I asked where Oliver found the Clostridium algae he showed. Now I found one next to my annelid. Yes. Yeah. If you find Clostridium, then look at the tips. Clostridium is not Clostridium. That's the dangerous bacterium. But Clostridium is a crescent, half moon shaped algae. And what you want to look is you want to look at the tips because there are these crystals that are kind of bouncing around in there. Yeah. So okay, I think it's already 1.5 hours, and I think I'm just almost going to call it a day. I'm just going to show you very quickly. Maybe not put it under the microscope, but how the jar looks like. So this is, remember about 2 weeks ago or so, I dumped everything into here. Yeah. And this is how it looks. Actually, there are still some water fleas in here. Okay. There actually are still some of them floating around. Maybe you see some dots, yeah? Okay, not quite as many as I expected. You know what I'm gonna do, just for the fun of it? Just for the fun of it, you know what? I mean, it looks a little bit cloudy still, so that means that there is still quite a bit of sediment, not sediment, of suspended material in here. Some, I don't know, some planktonic algae maybe or something. But you know, what I'm gonna do now is I'm going to, look, I show you what I've got here. Yeah. This here is yeast, and that is spirulina, which is compressed algae, which they're selling as a food supplement. I don't know why in the world anyone would eat this stuff. Yeah, but that's basically compressed algae. And I use this as a food. Look, I'm gonna put some yeast in here. A little bit of yeast so that the water fleas have something to eat. And from the algae, spirulina algae, what I do is the following. You want to hold it tight so that it doesn't fall in. You take some scissors and just scratch some of this stuff off. Okay, you see that this is powder falling down on the surface of the water. And that is now basically, yeah, going to last maybe a week. And maybe the water fleas are going to reproduce a little bit more. Yeah, because they like to eat yeast and whatever particles are in there. But I think they have plenty of food anyway in here, right? But I've been actually doing that, and then I was able to increase the number of water fleas and daphnia and whatever, and the ostracods and so on quite a bit. Yeah. So found the tips, filming them. Yeah, the tips yeah. Basically, I think they're made, or I read somewhere they're made of, contain the element barium or something like that. I don't know. Yeah, but in any case, those crystals on the tips of the Clostridium algae are quite well known. Yeah. Is spirulina under the spiral? No, I actually put it under the microscope once. It's at least in this compressed form, it does not look spiral shaped. Yeah. So that is yeah. So let me have a look if there are any other comments. At Microbehunter, I managed to get my large pink purple amoebas on camera doing binary fission. Oh, that's nice. Okay, thank you. Yeah, if you have any links that you want me to share with the community, please. Yeah. Okay. So I would like to thank Paul again for the donation. Okay. Thank you very much. And what I'm going to do today is I'm going to call it quits. Last comment, I cleaned some oxygen producing algae from the pond, squeezed the green water out, saved it, and packed with Clostridium and copepods. Oh, well, wonderful. Yes. Yeah, I think I'm just going to call it a day for today. Hope that you liked this stream. Again, yeah, please do subscribe if you have not done it already. So that's really important, not only to my channel, but also to other microscopy channels. Give a little bit support to the creators. And thank you again to Paul and to all of you for your questions and your contributions and everyone. Yeah, hope to see you again next week. I'll think again of a topic. Maybe I'm going to, yeah, why not do some recrystallization of some honey maybe, and or have a look now also at the other water sample. Maybe some more water fleas started to grow. We're going to see. Yeah, all the best. Happy microbe hunting as always, and see you next week again. Bye-bye.